give a short kind of summary. One of the things that worries me that I think was a little bit to the point that Mike Taylor had brought out this morning is we have to some extent, temper the enthusiasm of what we hear at these meetings with wanting to run home and start doing brainstem biopsies on everyone or signing everyone up for targeted therapy. I'm just going to begin by so I don't have any stocks, I don't have any ownership or employment with companies, but I do have a number of preclinical and advisory board meetings for getting drugs into trials. And what I'm going to present here are a number of results from a variety of different companies, and so I just wanted to make people aware of that. So what you've already seen so far with Stefan Pfister was this concept of the genetic alterations of low grades. He focused on pilocytic astrocytomas and then gave a little bit of information going forward on low grades to kind of set the stage about what the genetic landscape of these tumors are. And then Chuck kind of filled in how we use that knowledge of the genetic abnormalities to really allow us to move forward to begin to prepare some of the therapeutic interventions that might work and how we can actually begin to tailor some of those, recognizing that if we don't understand sufficient biology, we may choose the wrong drugs. But what I wanted to do was really begin to ask questions as practicing oncologists. How do we actually use this going forward into patients? What problems can we anticipate, because there are always going to be problems? And finally, where are the current standards? What should we be doing for our patients in the clinics now? Obviously, tempering both the enthusiasm with our relatively limited knowledge base on many of these therapies. So you've already heard of this concept of low-grade gliomas as a predominantly RAS-RAF disease. We knew this, in fact, long before much of the new molecular biology came out, just with our knowledge of tuberous sclerosis and the mutations associated with subependymal giant cell astrocytomas, as well as patients with neurofibromatosis, where we already see a very large percentage of pediatric low-grade gliomas. So we already knew this pathway was pretty important, but again, the work that Stefan presented from his group and others really highlighting the two major pathways, the V600E mutation of BRAF and the truncated fusion abnormalities, as well as a variety of others that he showed you that make up the almost 100% for this group of tumors. Certainly, I think, allows us to conclude that this really is a mostly single pathway disease. And the question is, can we now use this information going forward? Well, we've been using targeted therapies for low-grade gliomas longer than most people remember, originally with rapamycin, an mTOR inhibitor in subependymal giant cell tumors, given that we know where those abnormalities sit, as well as for now everolimus, which is approved in both Europe and North America for this patient population. And again, you see this kind of very interesting pattern of patients with these lesions that will respond to therapy. If you give them a drug holiday, the lesions grow back. If you start them back on drug, they respond again. And again, it's these kinds of things that remind us, and NF1 patients I think are very similar. This is not your typical malignant tumor where no matter what you do, they keep becoming more malignant and eventually die. This concept that you can stop and start therapy tells us that there's a unique biology here. And as Stefan pointed out, other than the BRAF mutations, we don't really see a lot else. And it may be that kind of pure single pathway disease that makes these behave a little bit different than what we're used to. We just completed a clinical trial through the Poetic Consortium looking at the mTOR inhibitor in sporadic low-grade gliomas. This is just an example of one of the patients that had some reduction in their disease. Certainly not every patient responds, but a number of them do for a therapy that's well tolerated. And this is a paper that's just about to be submitted for publication. So again, there are ways in which you can attack the pathway with already available compounds. I'm not going to show you the same results for the neurofibromatosis population. Again, we've just completed an everolimus trial in this patient population. There are still some patients on therapy, so the results are not ready to be reported yet. I can say that what's interesting is, in spite of the fact that every one of these patients starts the trial with exactly the same mutation, a knockout of neurofibromin, we don't see responses in all of them, we only see it in some of them, again suggesting that there are certainly other factors out there that have to be taken into account. And so although we're not seeing a second mutation that goes along with this pathway, there are probably some other factors that certainly do influence response, which means for the young people in the crowd, there are still lots of things to learn in this patient population. And then obviously with the utilization of some of the targeted drugs, this is dabrafenib, the V600E inhibitor. This is one of the clinical trials going on with the GSK compound. And again, you can see that this is a patient that has a 3.5 centimeter lesion that 8 weeks on therapy is now down to about 3 millimeters in size. And we're seeing these kinds of responses over and over, but again, only in patients with the V600E mutation. Now again, it's important to understand that there are only a handful of children in the world that have been treated with these drugs, and only for a matter of months, not years or decades. And given the outcome of this patient population, we need to kind of temper some of this enthusiasm with the reality of doing clinical trial in kids. In fact, as Chuck mentioned, this is an example of the Kaplan-Meier curve from the NYU group, where they did a BRAF inhibitor in an unselected population of kids with low-grade gliomas. And this is an unusual Kaplan-Meier curve that we normally don't see for low-grade glioma. Within 3 months, virtually all of the kids had had rapid progression, many of them tripling the size of their tumor. And very rarely do we ever see tripling of the size of a low-grade glioma within a matter of months. And it again gets back to the feedback loop that Chuck was talking about, where when you use a BRAF V600E targeted inhibitor in the truncated fusion, you actually stimulate feedback upregulation and therefore tumor growth, which means we have to be very cautious about who we select for the clinical trials and moving forward. And this is just an example of a patient with rapid progression weeks or months after initiation of that kind of therapy. So, and then finally, for those of you that were at ASCO a couple of weeks ago, was the first presentation of MEK inhibitor for progressive pediatric low-grade gliomas, in which 24 patients were treated, 8 of which had a radiographic response. So again, some new exciting information coming out. There are now 4 different companies with MEK inhibitors out there, all of which are competing. They have different advantages with respect to PK, blood-brain barrier penetration, and so figuring out which of these is going to be ideal, it's too early to say, but certainly that's a developing field. But in the context of all of this really exciting biology, what I didn't want people to do is kind of abandon what has served us, not ideally, but certainly well for the last 40 years, and that is that in spite of all of this increasing understanding of the pathways and the targeted drugs, is that we have an enormous amount of clinical trial data using more traditional chemotherapy, and we have 40 years of experience on which to understand morbidity from it. So you're not meant to read this. This is just a review article that we recently just published a couple of months ago that summarizes the huge amount of data for chemotherapy-based clinical trials for kids with low-grade glioma. And certainly, given that we have decades of experience, I think that for the time being, this continues to needs to be used, and that only through clinical trials should we be using some of those targeted inhibitors. Obviously, this is the most recent results of a randomized trial in the United States using either vincristine carbo or TPCV. I think we all recognize that kids with low-grade gliomas have frequent recurrences, so the time to progression or event-free survival is relatively poor, but we remind ourselves that the overall survival is excellent. And Astrid, in the next talk, is going to actually present the PSYOP data that I think will look very similar to this. But it is this issue here of understanding that because the vast majority of low-grade glioma patients are going to be long-term survivors, we have to be sure that as we introduce new therapies, that we are not a period of months, years, or decades down the road suddenly going to discover that we have done something that they would regret. And this is particularly important. Mimi Bandopattai and Peter Manley just published this SEER data of some 4,000 patients. And again, pediatric low-grade gliomas have an excellent long-term overall survival of decades, certainly compared to adult low grades, whether you take them all together or as pilocytics and just diffuse. And if you take patients once they reach the age of 23, basically their survival now is virtually guaranteed until they enter into the 50s and 60s and start dying of the stuff that we all start to die of. And so it's going to be very important that we take this into consideration. And Peter Manley is going to present some of the diffuse astrocytoma data from the SEER data set later on in the session just coming up, so I won't go into that in more detail. So not only do we have all of that chemotherapy and the toxicity data that goes with it, we obviously have a number of non-targeted but also non-chemotherapy-based therapies. And I just wanted to remind people that we're getting more experience with them, and therefore they still have to be considered as reasonable options. This is a metronomic approach that we've been using in Boston. This is a large bihemispheric tumor low grade for a patient that we never saw complete disappearance, but even over many years was certainly able to control its growth. And this has been reported in a number of different metronomic proposals. This is the data coming out of Toronto for the vinblastine therapy, again, where you can often see patients that have good prolonged responses in a therapy that's relatively well tolerated. Lenalidomide, a thalidomide derivative, again, doesn't have a particularly high response rate, but Kathy Warren is running a randomized trial of 2 different doses right now, where stable disease in the phase 1 trial was exceedingly high in this patient population, and again suggests that there may be opportunities there. And then finally, Roger Packer's group that looked at Avastin as a way of getting some control for these. So not quite targeted in the classic sense, but certainly things for which we already have now some years to a decade's worth of experience that need to be considered in the context of these diseases. So what I wanted to do is, just before we ended the symposium part and go to the lectures, is recognition that the landscape of pediatric low-grade gliomas is clearly changing and in evolution as we speak, and that many of the targets, predominantly in the RAS-RAF pathway, have now been identified, although there are still some variables that I think need to be identified. Critical issue is really going to be this concept of that as the clinical trials are underway, particularly for the targeted therapies, is that we have to remember these are investigational. They really should be done in the context of clinical trials. They require, as Stefan said, an understanding of the exact mutational profile of the patient. We can't just lump them all together. I think we learned that from the NYU trial, that we still don't really know the optimal dose, the schedule, the duration of therapy, the long-term or short-term toxicities of these, and before we kind of jump in feet first, we need to be aware of some of those as we kind of move forward. That given the number of available standard or somewhat newer therapies, figuring out how we're going to move these into upfront therapy, I think, is something that needs to be done in international collaboration between the groups, and that's therefore going to take a little bit of time. So it's one of those things where I want people to be excited about the biology, but not to kind of leap in, because as you can see, if you don't understand enough about, for example, the feedback loops, you can actually stimulate these tumors to grow, not inhibit them, and that clearly isn't our job. So I'm going to bring this, I just wanted to recognize this is the group from the Dana-Farber and collaborators that were part of this effort, and in particular many of the funding agencies, in particular Kids Brain Tumor Cure Foundation that supported this entire symposium, and as Chuck said, much of the work on which this was based. So I'm going to